Hello viewers, this is Ramakrishnan, Assistant Professor from the Department of Biochemistry, St.Joseph's College, Cuddalore. In today's program, I am going to discuss the clinical significance of enzymes, specifically lactate dehydrogenase. So, before to discuss this enzyme in detail, we will recollect the basic concepts about enzymes, isoenzymes, and clinical importance. Enzymes are the biocatalysts which can bind with substrate and forms enzyme substrate complex, then finally dissociate and gives the product. Generally, enzymes are all the proteinous substances made by amino acids. Enzymes plays vital role in the all metabolic pathways in our biological system, such as carbohydrate metabolism, amino acid metabolism, nucleotide metabolism, lipid metabolism, etc. Isoenzymes are a class of enzymes which carries a same biochemical reactions and gives same product, but the location of the enzyme or physical properties of the enzymes or molecular nature of the enzymes may differ, but product wise same. Clinical biochemistry is the branch of biochemistry which will discuss about the finding of a disease and the treatment and the way the treatment is progressing. All these things will be detected by finding the sample analysis, that is a particular compound from a sample. Samples are the substances collected from patient such as blood, urine, fecal matters, etc. So, from the sample, certain compounds will be tested, that is identified, both qualitative and quantitative study. From the findings, we are going to find the status of the disease, whether it is present or absent, or the severeness of the disease. That is called clinical biochemistry. Here we are using certain substances from the sample. Those are called marker agents. Here, enzymes act as biomarkers for various diseases or pathological situations. Under that, in today's work, we are going to select the enzyme lactate dehydrogenase. So, it is an isoenzyme which is essential one for the conversion of pyruvate into lactate in the presence of NADH2. So, lactate dehydrogenase is distributed in almost all the organs of our system, and specifically certain organs are enriched. For example, lactate dehydrogenase 1. This isoenzymes rich in cardiac muscles. LDH2 is rich in reticuloendothelial system. LDH3 is rich in lungs, and LDH4 is rich in kidneys, and LDH5 is rich in skeletal muscles. These divisions, that is LDH1, 2, 3, 4 and 5, are all carried based on the electrophoretic mobility of the sample from blood. Lactate dehydrogenase, when we look at the molecular nature, it is a tetrameric protein made by 4 segments. The each and every segment is made by 2 components. Either it may be a M segment or H segment. M means muscle and H means heart. So, generally, the molecular weight is 144 kilodalton. According to the different electrophoretic mobility, we have 5 categories. These enzymes and the subunits are represented by 2 colored images, green and red. So, green indicates that LDH H part, and M indicates red, that is, the red color is represented by the M. So, first LDH1, look at that, it is a tetramer of H, 1, 2, 3, 4 segments, and LDH2 3H1M, LDH3 2H2M, LDH4 3M 1H, and LDH5 all the 4 units are M segments. These are all separated by using electrophoresis. It is a separation technique based on the electric field. Since proteins are charged molecules, it will be separated based on the number of charges as well as in the molecular weight. So we will get a pattern like this. LDH1 will move in a faster rate than compared to 2, 3, 4 and 5. This is the ladder. This is the electrophoretic mobility direction. Generally, these enzymes are all intracellular enzymes. That means usually present in the inner side of the cell, so called intracellular enzymes. During normal situation, these enzymes are very, very low or not in the detectable range in blood. If there is any pathological condition, based on the location, these enzymes concentration will be elevated in the circulation and act as a marker for finding certain problems, for example heart problem or a problem in the lungs, etc. So that is called a clinical findings, that is act as marker enzymes. First, this image represents the LDH pattern for normal person. So LDH after electrophoresis, it is separated into 5 bands: LDH 1, 2, 3, 4 and 5. So generally in the normal case 2 will be higher in concentration, that is we will see a peak in 2. So normal level of LDH ranges from 140 units to 280 units per liter. In the case of any disease, we will see the change in this pattern. For example, in liver disease, look at this pattern. LDH5 will be in abnormal. The protein band will be very thick. So we will see a peak in LDH5. This is the pattern for liver disease. Whereas in the case of lung failure or any pneumonial attack, we will see the elevated level of LDH3. So that means thick band in the LDH3 part. And similarly, LDH1 will be elevated during heart disease. From this electrophoretic pattern, we will come to a conclusion that a person's sample is normal or abnormal. Say for example, heart problem or problem in the lungs or any other part. So based on their concentration of the individual fragments. All these things and all concluded after electrophoretic separation. That means the biological sample will be separated and the pattern indicates what kind of problem, whether it is normal or abnormal. So anyhow, these conditions are concluded with the other clinical findings also. Thank you.